Good afternoon. Thank you for joining us for the February 2020 CCC Spotlight Series. This month, we're highlighting Ilana Chilton, the point of contact, navigator, and genetic counselor for the VHL Comprehensive Clinical Care Center at New York Presbyterian and Columbia University Medical Center in New York, New York. Ilana is an American Board of Genetic Counseling certified genetic counselor who specializes in counseling for inherited pediatric and adult conditions. She received her master's degree in genetic counseling from Brandeis University and completed her undergraduate studies at the University of Pennsylvania. She currently works in the Division of Clinical Genetics in the Department of Pediatrics at Columbia University Medical Center, and she holds the title of Lecturer in Genetic Counseling and is co-director of the Foundations of Human Genetics course in the Columbia University Genetic Counseling Graduate Program. Ilana, welcome, and thank you for taking the time to talk to us.
Thank you so much, Josh. Thank you so much for that introduction, and thank you so much for having me today. So, as Josh said, my name is Ilana, and I am a genetic counselor at Columbia University Medical Center in New York City. And I want to tell you guys a little bit about myself, about our comprehensive care program here, and then I'm going to go into a little bit about the genetics of VHL, and then we'll do some questions and see what you guys have to say. So with that, I want to let you guys know that I've been a genetic counselor for about 8 years, and I've actually worked at Columbia for almost 5. As Josh said, I see both pediatric and adult patients in our outpatient clinic here, and I specialize in following individuals with rare and undiagnosed diseases, including VHL. So currently, one of my roles is to serve as patient navigator and genetic counselor for our VHL Comprehensive Care Center. And the sponsoring physician of the center is Dr. Wendy Chung. And I wasn't here when the program first started, so correct me if I'm wrong, Josh, but my sources tell me that we established our care center around 2005. So we've actually been serving the VHL community for almost 15 years now here in New York. And having a comprehensive care center at a large academic institution like Columbia really means that our patients have access to the highest quality specialists. And we're also fortunate to be surrounded by many other excellent academic institutions, which really give our patients a lot of options when it comes to finding the right specialists. And that's really important for people who have VHL, as many of you know. As I mentioned before, we follow both pediatric and adult patients in our care center, which means that we often see the whole family together when we see you at an appointment. And currently, we follow over 60 families with VHL, and we see these families on an annual basis. So we see them once a year. And as a genetic counselor and patient navigator, my job is to make sure our patients understand the genetics of VHL, that they understand the implications for not just themselves, but also their family. And I also work with patients to coordinate their annual screening, including imaging, blood work, specialist visits, and help them navigate any of the needs that they might have living with VHL. So I do want to highlight one thing, because as all of you know, coordinated care is extremely important for people with VHL, and it can really be difficult and frustrating to navigate our medical system when you have a rare disease that's unfamiliar to most medical doctors. As a care center here at Columbia, our goal is not only to educate our patients and families, but it's also to educate our community. And last spring, with the help of Josh in collaboration with the VHL Alliance, we held our first annual VHL Conference and Family Education Day here at Columbia. And this event was a full day event. It included presentations by Dr. Chung, several of our other medical specialists, and they provided a really nice overview of what we do here with VHL and what's sort of the latest and greatest in treatment for VHL. Overall, it was really a great success, and if you're local, we're really looking forward to offering this every year. So with that said, I'd like to transition over to opening up some of my slides and going through really the genetics of VHL with all of you. So give me 1 second and let me pop this up for you guys. All right, so I'm going to go through with you guys for about 10 minutes the genetics of VHL disease. Hopefully in doing this, I will actually answer some of your questions before you even ask them. But of course, I am happy to answer any additional questions that you might have. So, what is VHL disease? Well, as most of you know, VHL is an inherited genetic condition that is characterized by the formation of tumors and cysts in many different parts of the body. And most VHL tumors are benign, but some can progress to cancer if left untreated, especially those in the kidney and pancreas. VHL is an extremely variable disease, and because of this, it's often misdiagnosed and overlooked, especially by doctors who aren't familiar with the condition. VHL is caused by mutations in the VHL gene, but the real question that we have is, how do VHL gene mutations actually lead to tumor formation? How do they cause all the manifestations that we see in our VHL patients? So we can't really talk about the VHL gene if we don't talk about what a gene is. So I'm going to take you guys back to Biology 101 for a quick minute and explain that we all have cells, and in almost all of our cells, we have 23 pairs of chromosomes. And our chromosomes are these compacted forms of our DNA, and our DNA is a sequence of letters that actually codes for who we are. Genes are sections of our DNA, and these genes may code for things like our eye color, our hair color, our gender, and codes for things like how our body grows and develops. We're all born with 2 copies of every gene, and one comes from our mom and one comes from our dad. So how does cancer or tumor arise from changes in genes? Well, it arises from gene mutations. And the reason this happens is because some of our genes have the role of preventing our cells from growing and dividing too rapidly. So as I mentioned before, some genes code for things like our hair color and eye color, and other genes actually do things we can't really see. And one of those is suppressing tumor formation. If a tumor suppressor gene is damaged, or meaning it has a mutation, then it causes the gene not to work. However, because we have 2 copies of our gene, we have a second copy that's working, and our cell is fine. Over time, what happens is that our gene accumulates damage to the other copy, and now that cell has no working copies, and a tumor can develop because there is nothing telling the cell to not grow out of control. Why do these damages or mutations happen? Sometimes they're hereditary, meaning we're born with them. Other times they're caused by external factors such as radiation, smoking, or other exposures that can cause damage to our DNA. So this picture here is really showing you how a tumor develops in any person, because we're all at risk to develop tumors. But in a hereditary cancer condition like von Hippel-Lindau, what happens is that you are born with 1 damaged gene. And you are born with a damaged gene in every single cell in your body. And therefore, it only requires 1 extra hit to have 2 damaged copies and then lead to tumor formation. In the genetics world, we call this the Twohit hypothesis. So individuals who are born with 1 damaged gene have a much higher chance of developing tumors than individuals that have to accumulate these damages over time. So why are VHL gene mutations heritable? Well, VHL gene mutations occur at the point of conception and thus are present in every single cell in our body, including our germline, meaning our egg and our sperm. And because the mutation is present in our egg and our sperm, a person can pass this on to their offspring. In contrast, there is something known as somatic mutations, and these are mutations that occur in tissues after we are born and thus cannot be passed to our offspring. Cancer is actually maybe the combination of both heritable and somatic mutations, like in VHL. So VHL is inherited in an autosomal dominant fashion, and that means that 1 copy of the altered or mutated gene in each cell is sufficient to cause the disorder. As we learned, we all have 2 copies of every gene. Thus, a parent with a VHL gene mutation has a 50% chance of passing on the mutation to their offspring. Sometimes, though, we see that there is no family history of VHL. So if VHL is an inherited condition, then how can there be no family history? Well, the reality is that about 80% of the time, a person does in fact inherit that VHL mutation from their parent. But in about 20% of cases, VHL is the result of a de novo mutation. And what de novo means is new. And this is when a mutation occurs new to a person. These mutations are not present in the parents, but once they are in this person, then they can be passed on to future generations in the same autosomal dominant fashion. So what I'm really here to talk in more detail about is the VHL gene and why changes in the VHL gene lead to the manifestations that we see in VHL. Well, the VHL gene was actually identified many years ago in 1993 and is located on chromosome 3. It spans about 10,000 base pairs of our DNA. And as we discussed, our genes are a sequence of letters, and these letters code for who we are. The VHL gene actually provides instructions for making a protein that belongs to a class of proteins called tumor suppressors. So we talked about those tumor suppressors and how they're important in preventing a cell from turning into cancer. When there is a mutation in the VHL gene, it can either lead to a prevention of expression of the protein altogether, meaning no protein is made, or it can lead to an expression of an abnormal protein. Either way, the lack of protein or the abnormal protein actually results in promotion of blood vessel growth, and this is what is actually encouraging the tumor growth in VHL. The VHL protein is actually quite complicated and does a lot of different things. We don't have time to get into all of them today, so I really just wanted to highlight one of the primary functions of the VHL protein and how when that's not working, it encourages tumor growth. So one question that people have is, if I have a certain mutation in my VHL gene, will I have certain features of VHL? We're going to go into that, but in order to talk about the features of VHL, I wanted to just do a quick overview. I know that this is something that you all know, but just sort of head to toe, we know that vascular tumors called hemangioblastomas are the most common manifestations of VHL. They can occur in the brain, the retina, and sometimes they might be the initial manifestation in some individuals with VHL. We can also see cysts forming in the kidney and pancreas. Those cysts are typically not problematic. However, there can be a risk for either kidney cancer or sometimes for very rare neuroendocrine tumors of the pancreas. One of the other primary manifestations that we see in VHL are pheochromocytomas, and these are tumors of the adrenal gland. And very rarely we can see these endolymphatic sac tumors which occur in the ear. So when we think about those clinical features, the question is, and the question that many patients ask me is, well, if I have a certain mutation in my gene, will I only have some of those manifestations? And the reality is there are actually 200 different types of mutations that have been found in families with VHL. And there are many, many different types of mutations that can occur in many different parts of the VHL gene. So what you're seeing here is actually a diagram of the different parts of the VHL gene. We have exons in our gene, which are coding genetic information. There are other parts of our gene as well that we're not going to highlight. And this diagram is really just showing all the different types of mutations that can occur. Some of these mutations change single letters in our DNA, others cause loss of a few letters or extra letters, and some even delete the entire gene with all of the exons altogether. But because VHL is so variable and there are all these different mutations, a lot of the VHL research has focused on understanding if certain mutations can lead to certain manifestations of VHL. In genetics, we call this genotype-phenotype correlation. Thus far, about 4, sometimes we subdivide them into 5 categories have been identified. In general, we call these type 1 and type 2, but type 2 is further subdivided into types 2A, 2B, and 2C. As you can sort of see from this table on the left, every type is extremely variable. And that's really something I want to highlight in saying that we draw some genotype-phenotype correlations, but it's really important to remember that there is still variability. One notable correlation that we do see is that in individuals with type 1, there is a lower risk for PHEOs. Another notable correlation that we can see is that in individuals with type 2A, there was a lower risk for renal cancer, and in type 2C, there is actually no known association with renal cancer. Type 2C is predominantly, in fact, associated with strictly PHEOs, though other manifestations have been reported. So which mutations actually cause which type? Well, you can see on the bottom line of this diagram, really complicated words that explain the types of changes we see. Variants are also known as changes in genes or are mutations. And in type 1, what that's basically telling us is that these mutations lead to complete absence of the protein. So we talked about that sometimes the protein is totally gone and sometimes the protein is there, but it's abnormal. Type 1 leads to complete absence of the protein. In contrast, type 2 is primarily associated with what we call missense variants. Missense variants are when there is a single letter change that damages the gene, but protein can still be produced. What this tells us as researchers is that the type of VHL is somewhat associated with the type of genetic change rather than where the change is located in the gene. But there are still so many additional intrinsic and extrinsic modifying factors that lead to variability in VHL. And because of this, as clinicians, we really still recommend that you undergo screening for all manifestations of VHL, regardless of what type you have. So when we talk about screening for VHL, I'm not going to go through the details of it because it's not our focus for today. But I do want to say, especially at our center here, we have certain goals when we think about management and surveillance. And I think these are really the goals that the whole VHL community follows. And the idea is that because there are such a wide variety of features, that we really need to do routine surveillance, and routine surveillance allows for early detection, and early detection is key because early detection allows for timely intervention and improved outcome. The other thing with routine surveillance is it really gives us an idea of how many tumors are growing or changing or forming over time. And we know that with patients with VHL, the likelihood that they continue to form tumors is high. And so we really encourage the specialists that manage our patients with VHL to be cautious about intervening and to really only intervene when there are symptoms or when a tumor has a chance of metastasizing. We want to really minimize the number of invasive procedures that our patients want to have. If you follow the routine surveillance guidelines, we do see really good outcomes for our VHL patients. So as a genetic counselor, one thing that I'm obviously involved in is genetic testing for VHL. Just so all of you know, genetic testing for VHL can now be done on blood or saliva. It's actually a pretty affordable test. It used to be quite expensive, but most testing is now in fact less than $100. It's highly sensitive. And what that means is that 97 to 99% of people who have VHL will have a mutation in their VHL gene. Now, the flip side of that is that when I test you for VHL and you're negative, that means that it's extremely unlikely that you have VHL. And that's what a highly sensitive test is. So it's a very good test. So if you test negative for VHL, you likely do not have VHL. There are exceptions, but for the most part, that holds true. The other important element of genetic testing is that it can confirm a diagnosis and it can really help guide management. It helps us identify the individuals in the family that may be at risk or those that may not be at risk. And it can, for some people, aid in prenatal diagnosis and help make reproductive decisions. For each person, that's different, but it can be information that's used for reproduction. The other thing people often ask me is, do I need to test my children? And the answer is, in VHL, genetic testing for children is appropriate. And the reason that it's appropriate is because we can see manifestations in childhood in VHL. Specifically, we can see the PHEOs, we can see the CNS hemangioblastomas. We can see these things form, and we really want to make sure that we're screening for those. So typically what we recommend is by the time a child is 5 for you to consider genetic testing. We strongly encourage that the counseling be age appropriate. And we also note that sometimes identifying whether or not your child has VHL can cause anxiety. And so if one decides not to test, then we just recommend following the screening guidelines. So that's also an option. So I know a lot of questions that people have is, not what's currently going on with VHL, but, what is really next? And as I mentioned, we know that our patients who have routine surveillance have good outcomes. However, that doesn't prevent their tumors from forming. That doesn't treat VHL. It doesn't cure VHL. And so when we think about treating and curing VHL, the initial treatments focused on oral therapies, which would counteract that increased vascularization, which is what's leading to the tumor growth. And the hope of these oral therapies is that they actually shrink existing tumors and prevent new tumors from emerging. These are all still ongoing. I am not a specialist in any of these trials, but they are really interesting therapies that are under investigation. However, when it comes to genetics, what many people might ask, well, why are we focusing on drugs that slow tumor growth? Why don't we just fix this altogether? Let's just fix the gene. And the answer is yes, gene therapy is real, and this is absolutely coming down the pipeline. Just not yet, not right now, not today, but it will in the near future. And there are gene replacement therapies that actually exist and have been FDA approved for certain genetic conditions. And this is really a monumental breakthrough that I want to highlight, not just for those conditions, but for all rare diseases. Because these conditions who have already obtained FDA approval have really laid the foundations and the groundwork for what other rare diseases will need to undergo and the process that they will need to go through the FDA. So I think even though that's not VHL specific, it is really a notable advancement. What's challenging about gene therapy, though, is that it's really not a one-size-fits-all treatment, and it's actually quite complicated. As gene therapy, you might imagine, it must be gene-specific, it's sometimes mutation-specific, and depending on how you introduce the new gene into the body, it might in fact even be organ-specific. VHL has a lot of unique challenges in that it is a multi-systemic disease, and the gene itself is really involved in a lot of different functions and processes and pathways in our body. So the short of it is, we're not there yet, but gene therapy exists, and we are extremely hopeful that this will be a reality for VHL in the future. So with that, I just sort of want to highlight the different members of our team here at Columbia. Dr. Wendy Chung, again, is our sponsoring physician and head geneticist. I myself am the patient navigator and genetic counselor. Diana is our social worker, and she meets with all of our patients and families and helps coordinate our annual gathering. And everyone else there on our team is familiar with VHL, follows all of our VHL patients, and some are even involved in VHL research. So with that, let me go back to my screen and unhide it. I can figure out how to do that. Give me 1 second. All right, and I'm back. So Josh, if you wanted to go to some of the questions, that would be fine.
Yeah, thanks, Ilana. That was great. So we had a number of questions that were submitted in advance that people wanted to hear your thoughts on. So the first question is, why aren't pathogenic genetic mutations recognized during the DNA replication checkpoints and control points in the cell cycle?
So that's a really good question, meaning why doesn't our body just fix itself, right? Why can't our body repair those mutations when they happen? Well, the reality is that our body actually does repair the majority of mutations that our cells acquire, but because they occur at such a high rate, thousands and hundreds and millions and billions of mutations, the reality is that some of the mutations are simply going to be missed. And I know that's not an answer, a feel-good answer, but it's just that our body is imperfect, and statistically speaking, it can't fix everything. The other nuanced part of that is depending on the structure of certain genes, they are more prone to being mutated and less likely to be repaired. Again, we don't have time to get into the details of that, but that is one reason why we see recurrent mutations in genes that are not repaired.
Interesting, thank you. So before you spoke a little bit about the actual gene itself, so somebody asked, what exactly is an exon 3 mutation, and what are the implications of having this kind of mutation?
Excellent. You guys are asking such specific questions. I love it. So as I showed in my diagram, VHL actually has 3 exons. An exon is a part of our gene that is what we call our coding information. It's that sequence of letters that's going to code for a protein that's going to do a job in our body. All an exon 3 mutation is that your mutation is located in that part of the gene called exon 3. Now, when we talked about the genotype-phenotype correlations, we talked about the fact that where the mutation is located is not highly correlated with the type of disease. Rather, it's the type of mutation that you have that's more correlated with the manifestations that we would see.
Thanks. Why do some VHL patients develop malignant tumors while others develop benign ones?
So VHL is extremely variable, and I feel like that's sort of my word of the day. I've said it over and over. It is an extremely variable condition, even within a family. And the reality is that there are many different factors that actually contribute to a person's cancer risk. And a hereditary predisposition or a gene mutation in a gene like VHL is only one of them. So again, we talk about managing VHL. We also talk about healthy lifestyles, avoiding things that we know are carcinogenic or can lead to cancer. The other difference is that certain VHL-related tumors have a higher potential to become cancerous. On a whole, most VHL tumors are benign. However, there are the tumors in the kidney and in the pancreas do have a higher potential to become cancerous.
Great. I know you spoke a lot about types of VHL mutations. I was wondering if you could speak about where patients actually find their VHL mutation type, and is it something that would appear on their genetic report?
That's a really good question. This is not something that would appear on your genetic report. When you get your genetic report back, it's going to tell you the location of your gene mutation. And those are those P dot letters and numbers or C dot letters and numbers that we see on that report. And that's actually mapping out where in the gene your mutation is located and what type of mutation you have. Now, again, because these correlations are not foolproof, we don't actually report out what type of VHL you have. If you want to know what type of VHL you have, I recommend that you speak with your genetic provider and they can let you know if your specific mutation has ever been reported in association with one type. Again, I really do want to emphasize that regardless of which type of VHL you have, as clinicians, we still recommend that you screen for all manifestations of VHL.
Yeah. So I guess in VHL, there's always all these terms that are thrown around that can get very confusing. I was wondering if you could touch on the difference between Mosaic VHL and denovo VHL.
Yes, absolutely. So that's not actually, I didn't really address Mosaic VHL in my talk, so that is a really good question. And they're related, but they're different. So what I mean by that is mosaicism is actually when a person has 2 or more genetically different cell lines in his or her body. And this can happen in many different genetic conditions. And in VHL is actually quite rare. I think less than 5% of cases of VHL are mosaic. But the way mosaicism happens is that we all start as one cell, and this cell divides over and over and over again to create lots of different cell lines. If a mutation occurs at the beginning, then that mutation exists in every single cell line, and you have the same change in every cell. If a mutation occurs at a later point in that division, then some cell lines won't have that mutation, while other cell lines will. That's called mosaicism. All mosaic mutations are de novo, meaning they occur new. They're not inherited, because if they were inherited, they'd be in every part, they'd be present in every single cell. But not all de novo mutations are mosaic. Some de novo mutations occur at cell 1 and therefore are present in every single cell.
Thank you. That was a good explanation. So we just got a live question that maybe you could touch on.
Yeah.
Somebody said that they had a family member that had a mutation that was consistent with VHL, but the mutation has not yet been studied or identified. Is there a database with these less frequent mutations, or what are the next steps for a person in that situation?
So that's a really good question. To clarify, if they have a mutation in the VHL gene and they have symptoms of VHL, then they have VHL. But because the gene is so big, it's actually not uncommon that we see what we call private mutations, meaning that every family has a different mutation. Now, again, when we think about managing that type of VHL, we manage it all the same. So it doesn't matter which specific mutation it is or that it hasn't been studied before, we're still going to follow that person in the same way that we follow all people with VHL. But you make a good point that contributing to research is really important. All of the clinical labs that we work with actually contribute these types of results into shared databases so that they can be reported and studied. The other thing that can be helpful is that the VHL Alliance has a very good natural history study. So following the symptoms of VHL over time, and that will give us better information of whether you have a certain mutation that correlates with certain symptoms. So all of those things can be different things that you could consider participating in. But again, the bottom line is just because that change has not been studied before does not mean that we can't follow that person for VHL.
So I know you talked a lot about, or you touched on gene replacement therapy, how it's something that we could look forward to in the future. It's just not quite there yet. Where would somebody find more information about this type of therapy? And is there a difference between CRISPR and gene replacement therapy, or are they just different names for the same thing?
So in terms of, is there a difference between CRISPR and gene replacement therapy? There is. CRISPR is actually one laboratory method of altering DNA, and it is a tool that we use with gene replacement or for gene replacement therapy, whereas gene replacement therapy is actually a complete group of how we treat genetic diseases, and there are many different techniques that we can use when pursuing gene therapy, not just CRISPR. Where to go for more information on gene replacement therapy? The FDA is always has good information about what is current and the latest and greatest in gene replacement therapy. I would say anything VHL specific, the VHL Alliance should be your go-to. If you do want more specific resources about gene replacement therapy, then I would say check in with your local genetic counselor, your geneticist. You can email me and I'm happy to provide you with more links to read about it.
And I guess the last question that we have submitted is about, is there any research on gene therapy going on specifically for VHL, or is that a little ways out?
So that's a little ways out. I am not a researcher, so I am not in the lab. But again, there are a few hurdles that need to be jumped when it comes to gene therapy for VHL. And again, those have to do with the fact that the VHL gene and protein are involved in a lot of different pathways in our body, and the fact that tumors form in many different parts of our body. So the idea of where we would actually insert that gene is a tough question. So as far as I know, there is nothing yet, but again, I feel extremely hopeful for rare diseases that this type of thing will be coming down the pipeline.
And thank you so much, Ilana.
Yeah.
I just want to thank everybody who joined us today. I know we've had a lot of, we had some technical difficulties, but I'm so glad that you stuck with us. This was an amazing session. If you want to learn more about the VHL Comprehensive Clinical Care Center at New York Presbyterian Columbia, you can visit vhl.org/NYP Columbia. That's NYP COLUMBIA. This video will be uploaded and posted at vhl.org/Chilton, CHILTON. And again, I want to thank you, Ilana, for taking the time to talk to us today and for everything that you do for the VHL community.
Thank you so much, Josh. It's really my pleasure, and it really is a pleasure serving this community, and I hope to do it for many years to come.
Thank you so much, and have a great day.
Thank you, you too.